Mom, Dad, where do babies come from?
I think it's time we have the talk.
When a mommy and a daddy Arabidopsis plant really wants to have a baby, we undergo double fertilization to form an embryo. From there, the embryo develops within the ovary to mature into a dry seed so it can germinate whenever it wants to. Interesting. Can you tell me more about this process? How did I develop to become a seed?
Well, seed development is a multi-stage developmental process that includes two main stages, morphogenesis and maturation. It's important to note that this process as an entirety has hormones that contribute to the development and maturation of the seed, such as auxin, which plays a major role in elongation and makes sure the seed coat forms correctly. Cytokinins promote cell division and work together with auxin to control tissue development. Abscisic acid leads to seed dormancy and inhibits early germination. Ethylene controls the ripening and maturation of seeds and promotes seed coat thickening and the regulation of the embryo's shift from dormancy to active growth. Brasinosteroids regulate seed size and assist in the seed's adaptation to environmental stress during germination.
Morphogenesis is the initial stage for forming the seed structure. It's the development of the embryo, endosperm, and the start of the seed coat. Morphogenesis starts with the formation of the embryo. After fertilization, the zygote undergoes mitotic cell divisions. At the preglobular stage, cells are differentiated as the embryo proper and suspensor. At the embryo proper, the protoderm develops into the epidermal layer, the hypophysis develops into the root meristem, and the shoot meristem is also differentiated from the embryo proper region. At the globular stage, the ground meristem develops into storage parenchyma cells of cotyledons, and the precambium forms the vascular tissue in the heart stage. A depression is formed at the tip of globular structure, giving it the heart shape. This is the initiation of cotyledon differentiation. The elongation of the cotyledons has the embryo enter the torpedo stage. With more elongation and absorption of food material, the torpedo stage becomes a mature embryo. Meanwhile, during this process, two other things occur. The endosperm is cellularized, providing a nutrient source for the embryo. It is then consumed by the embryo, leaving the endosperm as a single layer that surrounds the embryo. The seed coat is also developed right after fertilization. The tissues surrounding the zygote start to develop and change into the seed coat. This happens by the integuments of the female ovary becoming the seed coat surrounding the embryo. During seed development, these integument layers change shape and thicken female reproductive organ and is hardened throughout maturation. Maturation is a stage where the seed undergoes growth and prepare for dormancy, ensuring that it can survive in various environmental conditions until it germinates. During the maturation process, the developing seed filial takes over control from the mother plant, maternal, and the embryo grows and the seed fills with nutrients. As the seed coat continues to take shape, the seed dries out of the end of the stage, which hardens the seed coat, serving a vital role in offering physical protection by becoming rigid. This enables the embryo to go into a latent state that lets it withstand a variety of environmental circumstances until it is ready to germinate.
After maturation, the seed's anatomy is made of structures that can ensure its survival and germination. These structures include external features such as the seed coat for protective outer covering, the hilum, the micropyle, which allows water absorption, and internal features include the embryo that contains the future plant, cotyledons, which store nutrients for early growth, and the endosperm that provides additional nutrients for the embryo.
But why is this important? What's the purpose?
Biological significance. Seeds are small but powerful. They are the natural way of ensuring how the plants survive, proliferate, and evolve in the natural world. Nutrient accumulation is one of the biological significance that take place during the seed developmental process. The accumulation of essential nutrients during seed development is a key factor that influences seed's quality and ensures its viability. Essential nutrients including protein, lipid, carbohydrate, and minerals are collected during seed maturation to promote the growth of subsequent seeding. Dispersal and colonization is another one of the biological significant in plant reproduction. Seed dispersal and colonizations are essential processes that enable species to form new population and adjust to various habitats. Genetic diversity, plant dispersion, and environmental stability are all influenced by these processes. Dormancy is another biological significance of seed development caused by internal factor and ensures germination occurs at the same time. At the right time, dormancy allows seeds to survive under unfavorable conditions. Adaptation and evolution is another one of the biological significance, which made it possible for plants to take over the land ecosystem. Each one of them provide a unique lens through which we can better understand the essential role seed plays in the natural world and how we see the life on Earth.
Oh, that makes sense. I didn't know seed development can be so important. Thanks, Mom and Dad.